Now we all use drugs recommended by doctors. We trust them without thinking about how they came to us. But do you know, every drug you and I use today goes through a clinical study before it reaches to the market. Yes, every drug that we get in the market today goes through a strongly regulated clinical study process wherein human participants are involved. These studies are conducted for two reasons. First, to check the safety and effectiveness of a new pharma product, and second, to expand the approved uses of an existing drug. When we say clinical trials involve human participants, it literally means drugs are tested on humans. That is why organizations developing drugs carry a serious responsibility. They must protect the safety of every participant involved. At the same time, they must prove that drug actually works, and this proof has to be demonstrated, which is why clinical trials follow strict procedures and why every step must be documented. Clinical trial audits are part of this larger system. They exist to verify that participant safety was protected and that the evidence supporting the drug is credible. But before we discuss what clinical trial audits are, what their objectives are, and when they are performed, it is important to understand why clinical trials are governed by such strong regulatory frameworks. There were a few critical moments in history, moments that changed how drugs are tested in humans forever. Some of these events were extremely tragic and they demanded a response. Let us take a step back and understand what these events were and how the healthcare and regulatory community responded to them. Events that shaped clinical trials. First is Nuremberg Code, 1947. The Nuremberg Code was introduced in 1947 during the Nuremberg Trials held in Germany after World War II. These trials were conducted to prosecute Nazi leaders and physicians for war crimes and crimes against humanity. Before this period, drugs and medical interventions were already being tested on humans, often without consent, without oversight, and often without accountability. There were no universal rules, no shared ethical boundaries. During World War II, Nazi physicians conducted medical experiments on prisoners. Prisoners were deliberately exposed to extreme conditions. Few of these experiments were high altitude experiments, freezing experiments, forced drug administration, infectious disease exposure. Many subjects suffered severe injuries. Many died. These experiments showed the world something alarming. Science, when unchecked, can become a tool for cruelty. There was global outrage when this came to light, but more importantly, the world realizes that ethical behavior in research could not be left to individual judgment or national discretion. There had to be a universal standard. The realization led to Nuremberg Code. It established the foundation for human rights in experimentation. The standards to set focused on voluntary consent, accountability for the impact of drug on society, clear evaluation of risk versus benefit, and other key principles that upheld the right of participants. Nuremberg Code was created in the context of war crime trials, and it laid the foundation to prevent abuse of human by researchers. It clearly puts across what must never be allowed in clinical studies. Second is Thalidomide tragedy and the Declaration of Helsinki, 1957 to 1964. In the late 1950s, a drug called thalidomide was marketed in Europe to treat morning sickness during pregnancy. At the time, clinical testing requirements were limited. The results were devastating. Thousands of children were born with severe limb deformities and congenital defects. These events forced the global medical community to rethink how human research should be conducted. In 1964, the Declaration of Helsinki, developed by the World Medical Association, was adopted. It laid down key principles that still guide clinical trials today. The principles were around free and informed consent, risk-based assessments, protection of vulnerable groups, use of placebos in post-trial access, ethical review, and environmental sustainability. Now that we understand the importance of ethical conduct and informed consent in clinical trials, let us move on to understand what clinical trial audits really are. So, what is a clinical trial audit? A clinical trial audit is a systematic, independent, and documented examination of trial activities. It confirms compliance, data integrity, and subject protection. If you come from a life science background, you already understand the seriousness of trials. These studies generate critical data that regulators rely on to approve a drug. They involve human subjects, which means safety, ethics, and accountability are non-negotiable in the process. And the trial must be controlled, well-documented process where every change is traceable. Audits are independently conducted to confirm that clinical trials meet these requirements. The objectives of clinical trial audits. Clinical trial audits are conducted with a few primary objectives: to independently verify that the trial was conducted according to approved protocol, to confirm that subject rights, safety, and well-being are adequately protected, to evaluate whether deviations were appropriately identified, documented, assessed, and corrected, to identify systematic gaps and process weakness that may impact compliance and data integrity, to assess inspection readiness and reduce the risk of regulatory findings. Let us look at the difference between clinical trial audit and clinical trial monitoring. Clinical trial monitoring. Clinical trial monitoring is an internal part of the clinical trial process. It is continuous in nature. It is done to keep the study aligned with the approved plan, whereas trial audits are conducted independently at defined point in the trial life cycle. Its purpose is to assess whether the trial conducted can be justified, reconstructed, or defended. Trial monitoring is done by a CRA or a monitoring team. They oversee site conduct, review data as it is generated, and addresses the issue before they escalate. Audits, in contrast, clinical trial audits are conducted by independent quality assurance auditors. They are not involved in trial execution. Trial monitoring. Clinical trial monitoring happens while the study is ongoing. It includes site visits, data checks, and issue resolution. Clinical trial auditing is more structured. It examines processes, documentation, and design trails. Monitoring happens throughout the trial, from trial initiation to closeout, whereas auditing happens at defined points before inspections, before database lock, or when risks are identified. Trial monitoring prevents clinical trial problems early. It improves data quality as the trial runs, wherein auditing verifies the trial conducted. It reduces the risk of data being questioned or rejected by regulators. Clinical trial audits. Focus areas. Clinical trial audits are aligned with global regulations and guidelines such as ICH GCP. Based on these requirements, audits typically review the following areas. First is trial conduct. Auditors evaluate how the study was actually conducted, then verify alignment with an approved protocol and study plan. They also assess how decisions, changes, and deviations were managed. Investigator sites. Here, the focus is on how individual sites conducted the trial. Auditors review informed consent, source documentation, and subject safety and protocol adherence as well. The goal is to confirm that sites protected participants and generated reliable data. Trial master file. Here, the audit is done to examine the completeness and organization of essential trial documents. Auditors check whether the TMF can reconstruct the trial from start to finish. A complete TMF is critical for inspection readiness. Data and systems. Here, the evaluation is done to see how trial data was captured, processed, and stored. Auditors review databases, access controls, audit trials, and data traceability. The objective is to confirm data integrity and system reliability. Types of clinical trial audits. For cause audits. For cause audits are triggered by a specific concern. These audits are conducted when there are serious deviations, data integrity issues, or safety signals. The aim is to assess the extent of the issue, determine its impact, and identify whether systematic failures are present. Inspection readiness audits. Inspection readiness audits are conducted before a regulatory inspection. They are performed when an inspection is expected or anticipated, like with pre-approval inspection, routine inspection, or for a cause inspection. The audit team stimulates how inspectors will review the data. Post-inspection audits take place after a regulatory inspection. They review inspection findings and assess systematic gaps. The goal is to strengthen compliance and prevent repeat issues. Follow-up audits. Follow-up audits verify whether corrective actions were effective. They check if identified issues were properly fixed and controlled. The goal is to confirm that risks have truly been reduced. With this, you should now have a clear understanding of what clinical trial audits are, why they exist, and the role they play in ensuring credible ethical drug development. If you are interested in more content on life science, clinical research practices, industry updates, and clear pathways in healthcare, follow our YouTube channel and turn on the bell icon to stay updated. Thank you for watching.
Visit us today, www.clinilaunchresearch.in.